Thanks, Tom. So I am from University of Illinois Springfield, and me and my students have been working out in the Tallgrass Prairie restorations at Emiquon for the last few years. So I'm going to be presenting some longitudinal data from that work. Oh, and I want to do a shout out to my co-author, which is Sarah Lindholm, who a lot of these data, the 2016 data, are all from her master's thesis. So I'd like to really put this in context. When we think about tallgrass prairie restoration, of course, that's a part of trying to stem biodiversity loss, right? So one of the things we're trying to do, whether we're with the Nature Conservancy or U of I, all of these different agencies, is to try to stop this kind of early stage of this mass extinction. So this figure is showing work that comes out of the Stockholm Resilience Center. And part of what they try to do is see where we are in terms of different environmental changes and whether we're kind of past this boundary of what they call high risk of Earth system destabilization, which sounds like something that would be really great to avoid. And what you can see here in this figure is that we've talked already about the importance of climate change in this symposium. And interestingly, other environmental changes actually get a kind of a higher ranking in terms of high risk from this group. One of those are our nutrient issues, right? So of course, people have already talked about nitrogen management and that type of thing. And then of course, the other thing is, of course, this very high rate of biodiversity loss. So when we think about where we are, where probably most of us are, right? We're here in central Illinois, and what did that region look like historically? Well, Illinois historically was almost all tallgrass prairie if we think about the percentage of the different biomes. And unfortunately, our tallgrass prairies have really been negatively influenced by land use, and to the point where they're actually considered a crisis ecoregion. So what these folks do to characterize which types of ecoregions are most at risk is they look and they see how much of that biome has been converted to another land use versus how much is protected. And if you have a really high ratio of conversion to protection, then it's a crisis region. And unfortunately, our grasslands often are characterized as crisis ecoregions because we have converted so many of them to agricultural lands. If we think about the tallgrass prairie specifically within the grasslands, you can see that historically, of course, that was most of what Illinois was, right, in terms of the ecosystem type. And that's also true for Iowa. So if you just look at the map on the left, you can see that pretty much complete cover in Iowa and really high acreage in Illinois as well. As opposed to if we look at the same coverage of tallgrass prairie in the 1990s, we can see basically the complete loss in these states where it's been really converted to agricultural land use. And the other thing is, we do know that we have some remnant prairie left, but they're just these very, very small remnants along railroads and things like that. So what do you do in a situation where you have an ecosystem that you want to preserve, right? And yet you don't have it there anymore. So preservation is not an option. What you do then, of course, is restoration. So that gets us to restoration ecology. Emiquon, of course, primarily is a floodplain restoration, but there are also over 360 acres that were put into tallgrass prairie because they were more upland sites. So this shows the prairie seed packets, right? These very big prairie seed packets, right, that are going in to do the Emiquon seeding restoration. So just to orient folks in terms of where the Emiquon Preserve is, if you're tuning in from somewhere else, basically located in central Illinois along the Illinois River, you'll see the photo that is here is actually the same as what I'm using for my virtual backdrop, and it's the bluff at Emiquon looking out over those floodplain lakes. This shows you the map of the preserve. Basically, the light green here is the Emiquon Preserve itself, and then in yellow you can see those are the Tallgrass Prairie units. And specifically, we've been collecting data for these 5 Tallgrass Prairie units that you see over on the right, and those are their management unit numbers. And you don't need to memorize those or anything, but I will be referring back to those later, because one of the things you'll see is that over the time period we've been collecting data, different disturbances have affected those different units. So some of the basic questions we're interested in are just, one is just a very general question of what patterns of change in the plant community are we going to see over time. So it's kind of really a luxury to be able to go out to the same locations at various points in time and see how the community shifts. So just what are those general patterns of plant succession? And then, of course, we're also interested in, well, how do we gauge that? How successful has that restoration been, right? So we know that we want to try to preserve Illinois' biodiversity. How successful are we in terms of putting in this plant community, which hopefully will then impact the insect community, bird communities, all of that. So for all of these sites, we have at least these 2 years of data. We have longitudinal data, first right after they were originally seeded, so in 2008, and then the next was in 2016. And for a couple of the sites, we also have some 2012 data as well. And this is kind of a gold standard to have longitudinal data as opposed to be looking at prairies of different ages to try to figure out changes, right? So we're going back to the exact same locations. So what exactly was seeded and what has the management been in these units? So along with all the rest of the Emiquon restoration, this site was seeded or started to be restored in basically the field season of 2007, right? So April 2007, and then that January, that should be 2008. And so over that field season, 81 species of native forbs and grasses. And the thing for these management units is they had identical seed mixes in all of the units. They have been managed since that initial seeding with herbicide spot treatment of invasives. And they have been burned, although with different frequencies in the different sites. So all of the sites were burned early on in either 2008 or 2009. And then 3 of the 5 sites were also burned in 2015. That's sites 16, 17, and 22. So here you see some of the field crew, happy people going out and collecting data on seeds and seedlings. So what we've been doing is going back to these same transects at different periods of time. Now, so the exact plots that we sample along the transects change, but the actual transect location is the same. And what we've been doing is doing what's called a floristic quality assessment. So basically going in, setting up, you can see these 1 meter square plots that are, the 2 transects are 50 meters apart and the plots are 10 to 20 meters along each transect. And then we go in there and we basically identify all of the typically herbaceous plants. So all the vascular plants that are in each 1 meter square plot. Again, we have data for all of these sites for 2008 and 2016. And then for 2 of the sites, 17 and 22, we also have an intermediate year in 2012. And for each of the sites, we have 15 plots, so 15 1 meter square plots to make 75 plots total. And actually this plot size of 1 meter squared, for those, not to get too in the weeds, but for people who want to do plant measurement and sampling, this is actually a larger plot than a lot of people have used historically, but it actually has been shown to be good for lowering the variation in mean C, which is one of the metrics I'm going to be talking about. So it's actually a good plot size for that. Now, you note that time period, 2008 to 2016, as Mitch was already saying, we've had a lot of kind of unanticipated disturbances along the Illinois River over that time period. And for these management units, the most specific thing was this large historic flooding event that happened in 2013 when there was overtopping of the Illinois River, and those river waters actually ran into 3 of the management units. So these are the 3 prairies that were affected, the site 16, 18, and 19. You see a photo here of just the overtopping of that channel that then drained down into some of those management units. This is just showing a different perspective for a couple of the different units. Again, this site 18, now you can see this body of water that was actually new starting in 2013. So when we had sampled previously, that hadn't been there. And then after that 2013 historic flood, a whole new water body at the southern edge of 2018, and at the southern edge of site 18. And then in site 19, you can see the debris that was brought in with those floodwaters. So we have 5 different sites, but what's interesting is that they experience these different levels of disturbance and also different types of disturbance. So we have those 3 sites that had the flooding, and then we also have the other 2 sites, site 17 and site 22, which didn't get flooded and also were burned twice. And you'll also note that 2 of the sites that got flooded hadn't had another burn in 2015. So we have 5 sites with different disturbance histories. So again, the data that we're collecting are floristic quality data. These are based, this method has been around for decades now, developed by Swink and Wilhelm. And the main thing you kind of need to kind of track some of these metrics to understand is this idea of the C value. And I'm just going to call it a C value because it's fewer syllables, right? So any given plant can be given a coefficient of conservatism of anything from 0 to 10. 0 are things like our non-native invasive plants, or 1 for a native weed, and then all the way up to a value of 10 would be those plants that are really demonstrate high quality, are only kind of found in high quality prairies. So as an example, I've shown this picture of the compass plant here, and it has a C value of 5, so a fairly high C value. It's kind of an iconic species for the prairie. Now, the metrics I'm going to show, we looked at them at 2 different spatial scales, so you can either lump all your data together, right, for a site, and those are the cumulative data, and look at that species list and calculate these metrics, or you can do it at the plot scale. So the metrics we look at to assess quality, one is just that mean C value, right? So of course, as the quality increases, that average C value will also increase. Another metric is floristic quality index, which is similar to the mean C, except it's also includes the native richness. So you count up how many native plants you have, right? And based on that, you take the square root and you get your FQI. And then of course, just the regular richness as well. And because of the flooding, we also looked at wetness values, right? So we were interested to see whether the plant communities would shift in that respect. And wetness values just range from your upland plants will have a high value of around 5, and then your obligate wetland plants will be a -5. Okay, so the big take home of what we found is that these disturbance histories correlate with the restoration outcomes. So those two sites, 17 and 22, that had burned twice and not flooded, we're seeing improved floristic quality in those sites, as opposed to the 3 sites that hadn't been burned as frequently and were flooded. Those are declining in quality. So this shows you the data for the 2 sites. These are the cumulative data for the 2 sites that, again, were burned twice, but not flooded. And you can see that increase in FQI, and then also that increase in the mean C, as well as over on the right, the increase in the native richness for the site, and the kind of not really decrease, but kind of no real problems with non-native invasion, as opposed to the sites that, well, 2 of them were only burned once, and then also they experienced that flooding. And what you can see in those sites is that the mean C did not increase. The FQI for a number of them actually declined. And same with the native richness. At least for 2 of those sites, we saw a decline. Now, one thing that's interesting, that site you see in orange is actually the site that was burned in 2015. So perhaps that's some evidence that kind of burning might help in terms of resilience from flooding impact. So basically, we see this difference in the restoration trajectories depending upon disturbance history, and what that is causing actually is an increase in variance in these floristic quality measures over time. So if we actually look at 2008 and we compare it to 2016, and we look at the mean C values, we see they're more variable now, right, or in 2016 than they were back in 2008. And so that kind of is consistent with this idea that these sites are kind of going in different directions, if you will, with some improving in quality and some not. This is just data at the smaller spatial scale. We don't have a lot of time, so I'm just gonna say we basically saw the same thing at the smaller spatial scale. And we also saw correlates with the wetness. So specifically, higher floristic quality was correlated with having more plants that were kind of had that upland type of a wetness coefficient. Okay, so what does that mean in terms of restoration success? Well, one thing in terms of KEAs and all of that for Emiquon, I think using a mean C would be a great KEA for Emiquon to do, in part because unlike some of these other metrics I've talked about, it's really more resistant to changes in sampling effort, right? So you'll get a pretty good estimate of mean C at different levels of effort. And so if we look at our mean C values that we see for 2016, you can see, again, those 2 sites, the two that got burned twice are in that realm of having high floristic quality, whereas the other 3 are lower, and they're not kind of meeting that mean C that we'd like to see of at least 3.5. Okay, so I've kind of already said this, the restoration trajectories differ among the sites. It seems like burning is good in terms of enhancing the floristic quality, whereas the flooding appears to have degraded it. And what are we going to expect in terms of our restorations, in terms of quality over time? You know, it really depends on the study you look at. Some really definitely show improving quality over time. Hopefully we'll continue to see that at Emiquon, but it may really depend on disturbances. And then I think the other big thing, and when we think about management, is there anything that we could have done to kind of prevent that decrease in quality despite the flooding? I think that's hard. I think the only thing I might recommend is just having a few more plants in the mix, right, that had that those lower wetness coefficients, but also were high in terms of their C value. So kind of maybe have more of that mix. And that's it. So I think hopefully I have a minute or two left to be able to do questions. I don't want to go into the break time. I do want to do a big thanks because it's always never just me, right? So again, Sarah was a co-author. Christy and Emily also had data that from their master's thesis, and then all of the people who help us in the field.
All right, thank you very much, Amy. That was great. We do have a couple minutes for a question or two. I know one just came in, so I'll turn it over to Doug.
Thanks, Tom. Thank you, Amy. First question that we received, how does burning affect pollinators like mason bees, etc.?
Yes, I don't know about pollinators, and as far as I know, I'm not sure if we've collected pollinator data. I do know that some folks, what they would recommend is that you just burn half of the unit, right, at any period of time, right, to kind of protect some of your pollinator populations that might be in the stems and things like that.
Good, and we've tried to do that, not burn all the sites in the same season, the same year. So hopefully that provides a place for at least some of them to escape to.
Right.
Here's another question. Water can transport seeds and other plant propagules. Do you believe that any new species showed up at the sites that had been flooded that could have been brought in by the floods?
Maybe. I mean, we definitely saw a lot more weeds like velvet leaf and stuff like that afterwards. But they could have very well been in the seed bank anyway. It's hard to know. And we didn't, I know I had that thought because I used to do seed dispersal, like, oh, I should be out there getting the water and seeing what's coming in. But we don't really, there's no plant, like a rare plant that I don't think wouldn't have been in the landscape anyway that we really saw coming in.
No lotus or anything like that we've seen.
Yeah. Nothing that wasn't there otherwise, even before the flooding. Yeah.
Good.
Thank you, Amy. Thank you.